अस्सलाम वालेकुम students, तो हम डिस्कस कर रहे थे द hallmarks of the cancer, in which the second one is insensitivity to the growth inhibitory signals. Insensitivity means that the cells stop responding to the growth inhibitory signals, and there will be no inhibition of the cellular proliferation, and the cell proceed towards the new plastic growth. And the growth inhibitory signals, they come from the tumor suppressor gene. Normally, the tumor suppressor gene, they control the cell cycle, and they decreases the cellular proliferation and the DNA replication. So the tumor suppressor genes are involved in regulating the transition of the cell from the G1 phase to the S phase. If there is any damage to a DNA, then there will be arrest of the cell cycle occur at the first checkpoint. Similarly, the tumor suppressor genes are also involved in the DNA repair, and the DNA repair occur mostly through the P53 gene. Similarly, the tumor suppressor gene are also involved in regulating the inhibition of the mitotic pathway, and the mitotic pathway is inhibited through the APC gene, neurofibromine 1 and 2 gene, and the P10 gene. And the tumor suppressor gene also causes inhibition of the angiogenesis, and the process of angiogenesis is inhibited by the von Hippel-Lindau genes. And the various kind of tumor suppressor genes include RB genes, P53 genes, APC genes, E-cadherin genes, neofibromine 1 and 2 gene, and the P10 genes, transforming growth factor beta genes, WT1 genes, and the von Hippel-Lindau genes. Normally, the tumor suppressor gene, they inhibit the cellular proliferation, but in neoplasia, there is loss of function mutation occur in the tumor suppressor gene, and the mutation in the tumor suppressor gene may be sporadic or they may be familial. Sporadic mutation occur in about 60% of the cases, while familial mutation occur in about 40% of the cancer. So the first one is the sporadic mutation. A sporadic mutation is the acquired type of mutation in which the both alleles are mutated after the birth. Normally, these alleles are normal at birth, but the mutation occur in both alleles after birth. The second one is the familial pattern. In familial pattern, one allele is mutant at birth, but the second allele is normal. So we said that there is existence of the heterogeneity, but with time there may be mutation occurring in the second allele. And when both alleles of a gene become defective, then we say that there is loss of the heterogeneity occur. And the tumor suppressor gene, they shows recessive pattern. In recessive pattern, both allele of a gene are inactivated or they are defective. अब बात करते हैं about the Knudsen two-head hypothesis. It is also known as the double head hypothesis. And according to this hypothesis, both the alleles of a gene should be inactivated or mutated for the development of the cancer. In the first head, this mutation occur in the first allele, whereas in the second head, the second allele also become mutant. So, according to the Knudsen two-head hypothesis, for the development of cancer, both the alleles of a gene must be inactivated. अब आ जाते हैं about the various types of the tumor suppressor gene. The first one is the RB gene. RB gene is the first tumor suppressor gene to be discovered, and initially the RB gene was found in the retinoblastoma of the retina, but later on it was found that this gene is present in all the cell. Normally, the RB gene is present on the chromosome number 13 Q arm at the area 14, and the RB gene is known as the governor of the cellular replication. In normal cell, when there is increase in the growth promoting signals occur, and the MOSC gene, they activate the cyclin genes to produce cyclin, and the cyclin combines with the cyclin dependent kinases and they form a complex, and these kinases, they phosphorylate the RB gene. Normally, the RB gene, they hold the E2 vector and prevent it from releasing, and hence they prevent the process of the cellular proliferation. But when the kinases enzyme, they phosphorylate the RB gene, the RB gene become inactivated on phosphorylation, and the E2 vector is released, and this E2 vector then causes the DNA replication. So on phosphorylation of the RB gene, the RB gene become inactive and they releases E2 vector which causes the cellular proliferation. But if there is an increase in the growth inhibitory signals occur, then these growth inhibitory signals inhibit the formation of the cyclin-dependent kinases and cyclin complex. And when there is no complex formation occur, then there will be no phosphorylation of the RB gene. And when the RB gene is not phosphorylated, it hold the E2 vector, and there will be no release of E2 vector occur and no DNA replication. When the RB gene is present in the hypophosphorylated state, we say that the RB gene is in active form and it holds the E2 vector. But in neoplasia, the RB gene become mutant and there is loss of function mutation occur, and it follow the principle of Knudsen 2 hypothesis. It means that both the alleles of a gene are mutated in the RB gene, and when there is mutation in the RB gene occur, it leads to the development of the retinoblastoma. And the retinoblastoma is the most common malignant tumor of the retina. And the retinoblastoma may be familial type or the sporadic type. And familial type, the one allele of the RB gene is mutant at the birth, and the second allele become mutated after the birth. And the familial type of retinoblastoma are usually bilateral, means that they involve the retina of the both eyes. And the sporadic retinoblastoma, both the alleles become mutated after birth. And the sporadic retinoblastoma is usually unilateral, means that it involves the retina of one eye. Similarly, there are also certain oncogenic viruses like Epstein-Barr virus and human papilloma viruses, which releases the protein known as E7 protein. And this E7 protein, they inactivate the RB gene, they hypophosphorylate the RB gene. So when the RB gene is inactivated, there will be release of E2 factor occur, which causes increase in DNA replication and the cellular proliferation and leads to the new plastic changes. So the second one is the p53 tumor suppressor gene. P53 gene is known as guardian of the genome. It is also known as the molecular policeman of the genome. And the p53 gene consists of three forms. The first one is the wild type, second one is germline p53 mutation, and the third one is sporadic p53 mutation. The wild type p53 gene is found in the normal cell and it is present on the chromosome number 17. The germline p53 mutation in which one allele is mutant at birth and the second one become mutated after birth, and it causes Low-Perimi syndrome, which is associated with various malignancies like leukemia, sarcomas, breast cancer, and the brain tumor. And the third one is the sporadic p53 mutation, in which both alleles of p53 gene become mutated after birth, and it is the most common genetic mutation seen in the human cancer. In normal cell, the p53 gene they binds with the MDM2 gene, and the MDM2 gene they degrade the p53 gene and reduces its half-life. The p53 genes they control the cell cycle at both the checkpoints from G1 phase to the S phase up and from G2 phase to the M phase. If there is any damage to a DNA, then there will be arrest of the cell cycle occur at both phases. There are certain DNA containing viruses like human papilloma virus, which releases a protein known as E6 protein, and this E6 protein causes inactivation of the p53 gene, and which causes cancers like genital cancer. Normally, if there is increase in the stress occur on the cell due to ionizing radiation or the carcinogen, they causes damage to the DNA, and the damage to the DNA is sensed by certain factors like ATM and the ATR factors. ATM stand for ataxia telangiectasia mutant factor, and ATR stand for ataxia telangiectasia with 3 protein. And these ATM and ATR they sense the DNA damage and they activate the p53. So the activated p53 gene activate p21 gene, and the activated p21 gene causes inhibition of the cyclin-dependent kinases 4 and 6. And when this cyclin-dependent kinase is inhibited, then there will be arrest of the cell cycle occur at the G1 phase. Similarly, activated p53 gene they activate GAD45 protein, which causes the DNA repair. And the activated p53 gene also activate the apoptotic genes like BEX and BEC family, which causes the apoptosis. So the activated p53 gene they arrest the cell cycle at the G1 phase, and when the damage is less, then they causes repair of the DNA. But if there is more damage to a DNA, then p53 gene induces the apoptotic gene and causes apoptosis of the cell. But in neoplasia, there is mutation in the p53 gene. Again, the mutation is loss of function mutation, which shows the recessive pattern, means that both allele of p53 will be mutated. So when p53 gene is mutated, then there will be no arrest of the cell cycle occur, no repair of the DNA occur, and there will be no apoptosis occur, and the cell become immortal, and there are more mutation accumulating in the cell with time, which leads to the malignant changes. Similarly, if there is damage to a DNA occur, then there will be activation of p53 gene, and when the p53 gene is activated, it binds with the damaged DNA, and it causes the activation of the microribosomal 34 family, and the microribosomal 34 family is a type of the microRNA, which causes inhibition of certain gene like they inhibit the growth promoting genes like MYC gene and cyclin dependent kinases 4. And when these genes are inhibited, there will be no cellular proliferation occurs. Similarly, they causes inhibition of the anti-apoptotic genes like BCL2. When the anti-apoptotic genes are inhibited, there will be the apoptosis of the cell occur. So the P53 gene either causes arrest of the cell cycle and they repair the DNA, and when the DNA cannot be repaired, it causes apoptosis of the cell. Similarly, they cause inhibition of the growth promoting gene, so there will be no cellular proliferation occur, and they also inhibit the anti-apoptotic gene, so they induces the process of the apoptosis. The third one is the APC gene and the beta-catenin pathway. APC stands for adenomatous polyposis colonic gene. In normal cell, the APC gene they codes for the APC protein, and this APC protein combines with the beta-catenin gene and they inhibit the beta-catenin gene. Normally, the beta-catenin gene is activated by the WNT gene, and when the beta-catenin is activated, it activates the MYC and the cyclin gene and causes the cellular proliferation. To prevent the overactivation of beta-catenin, the beta-catenin is inhibited by the APC gene. So the APC gene causes inhibition of the beta-catenin gene. And if there is mutation in the APC gene and the mutation is loss of function mutation, then there will be no inhibition of the beta-catenin gene, which causes the activation of MYC and cyclin and leads to all the various new plastic changes. And the APC mutation is more common in the adenomatous polyposis colonic cancer. So the next one is the loss of contact inhibition. Normally the cells are attached with each other to the E-cadherin, and the E-cadherin they hold the cell with each other. And similarly, when the E-cadherin they expresses, they bind the beta-catenin gene with the APC gene and they inactivate the beta-catenin gene. So the E-cadherin they control the activity of the beta-catenin gene and prevent it from overactivation. If there is mutation in the E-cadherin, then there will be a separation of the cell occur. Similarly, the activity of beta-catenin cannot be inhibited because the E-cadherin is mutant, and the beta-catenin will overexpress and will causes more cellular proliferation and leads to all the new plastic changes. The other tumor suppressor genes include P10 gene, which is a tumor suppressor gene, and P10 gene has a phosphatase activity, and this gene is involved in controlling the PI3 kinase pathway and the AKT pathway, which are involved in the cellular proliferation. And if there is loss of function mutation in this P10 gene, it causes endometrial and the prostatic cancer. The next one is the BRCA1 and BRCA2 gene. These BRCA1 and BRCA2 gene are the tumor suppressor gene, and they are involved in the DNA repair. The BRCA1 gene is present on the chromosome 17, and the mutation in BRCA1 is associated with the breast cancer and the ovarian cancer. And the BRCA2 gene, which is present on the chromosome number 13, they are involved in the breast cancer and the prostatic cancer. The next one is the neopibomin 1 gene. The neopibomin gene, 1 gene is a tumor suppressor gene, and it is present on the chromosome number 17, and this gene code for the new pibomin 1 protein, and this new pibomin 1 protein has a GTPase activity, and it is involved in regulating the activity of the RAS gene, and the new pibomin 1 gene they control the overactivation of the RAS gene. If there is mutation in new pibomin 1 gene, it causes new pibomatosis type 1. The next is the new pibomin 2 gene, which is present on the chromosome number 22, and if there is mutation in this new pibomin 2 gene, it causes aqueous neuromas, CNS tumors, and the lesh nodules. And similarly, there are also cupulated spots occur in the new pibomin 2 gene mutation. The next tumor suppressor gene is the WT1 gene, which is a tumor suppressor gene present on the chromosome number 11. And the WT1 gene is involved in the mesenchymal to the epithelial transition. So if there is loss of function mutation in the WT1 gene, it will lead to the development of the Wilms tumor, which are the tumors of the kidney. And Wilms tumor, there is no differentiation of the nephron occur. And the next tumor suppressor gene are the INK4A gene and the ARF gene. The INK4A gene they blocks the RB phosphorylation, and the RB gene is present in the ectoplasm, and it holds the E2 vector and prevents the cell from the replication. But if there is mutation in the INK4A gene, then there will be a phosphorylation of RB gene and E2 vector is released, which causes the new plastic changes. And INK4A mutation is involved in pancreatic cancer, breast cancer, and the esophageal cancer. whereas the AIF gene, which is a tumor suppressor gene, and they prevent the p53 destruction. But if there is mutation in the AIF gene, it will lead toward the various bladder carcinomas, head and neck cancers, and the leukemias. The next tumor suppressor gene is the von Hippel-Lindau gene. In normal cell, when this gene expresses, they produces protein which causes degradation of the hypoxia-inducible factor alpha, and the concentration of this hypoxia-inducible factor decreases, which causes decrease in the synthesis of the vascular endothelial growth factor. So there will be no angiogenesis occur, and there will be a decrease in the cellular growth occur. But if there is mutation in this gene, then the concentration of hypoxia-inducible factor will increases, and there will be no degradation of this factor occur, and hence it will lead towards a more angiogenesis and increase in the cell growth, which leads to the new plastic changes. And the von Hippel-Lindau gene mutation is present in the cerebellar hemangioblastomas, peuchromocytomas, retinal angiomas, and the renal cancer. The next tumor suppressor gene is the transforming growth factor beta, which is a tumor suppressor gene, and the transforming growth factor beta, they bind to the receptor and activate the receptor. And this receptor then causes intracellular signaling to the SMAD2 and the SMAD4 factor. And when these factors are activated, they causes activation of the cyclin-dependent kinase inhibitor. And when these inhibitors are activated, they causes inhibition of the cellular proliferation and the DNA replication. So the transforming growth factor beta, they inhibit the cell proliferation through the activation of cyclin-dependent kinases inhibitor. And when there is mutation in the transforming growth factor beta, it will lead to the cancers like pancreatic cancers and the colonic cancers.